I'm going to repeat that. These seminars are offered free of charge thanks to a workforce development grant received by the University of Oklahoma Department of Psychiatry and Behavioral Sciences from the Oklahoma Department of Mental Health and Substance Abuse Services. Each Friday in September through May from 2 to 3, with the exception of winter break and holidays, we gather here to listen to a presentation on topics related to the understanding and treatment of addiction and chemical dependency. Thank you for attending the seminar. Now I would like to introduce our speakers. Doctors Jean Halford and Danielle DeMarco. And Dr. DeMarzo, I think I pronounced that wrong the first time, it's DeMarzo. In 2002, Dr. DeMarzo graduated from the University of Miami with a degree in psychology, focusing on neurosciences and double minors in biology and chemistry. Her undergraduate research was in bipolar disorder and factors influencing compliances with medication regimens, as well as how life events trigger depressive or manic episodes. After earning a medical degree with honors from Ross University, she moved to Oklahoma in 2007 for residency. She completed residency training in obstetrics and gynecology in 2012 at the University of Oklahoma School of Community Medicine in Tulsa. As a genetic technology, as genetic technology advanced and became more available to provide diagnosis for her patients, she left her private practice in 2016 to further her training. She returned to the University of Miami for fellowship in clinical genetics. She returned to Oklahoma and has been practicing in Oklahoma City at the OUHSC since completing fellowship in 2018. She has interest in cancer reproduction genetics, but her day-to-day practice focuses on pediatric diagnostic practice. Dr. DeMarzo Birdsall has authored or co-authored and peer-reviewed manuscripts in 5 published abstracts and poster presentations at the ACMG and other national and regional conferences. While working full-time in the section of genetics, Dr. DeMarzo Birdsall provides a full range of genetic services in her clinic. In addition to providing her clinical care, she provides teaching for the Masters of Science students in Genetic Counseling program, the Pediatrics Residency Program, the Medical Genetics Residency Program, and the Combined Pediatrics Genetics Residency Program, and the MFM Fellowship Program. She also teaches medical students in both clinical and preclinical trials. Dr. Jean Halford, in the fall of 1989, began his career at the University of Oklahoma Health Sciences Center as a research assistant at the Oklahoma Center for Alcohol and Drug-Related Studies. For most of the next 14 years, he worked for Doctors Oscar Parsons and Sarah Jo Nixon in the Cognitive Studies Laboratory. Initially, he worked on several studies looking at the impact of chronic alcohol and in cognitive and psychophysiological performance. Over time, his work moved into the areas of dual diagnosis, working particularly with substance abuse patients with psychiatric disorders. Following this line of interest, he undertook and completed his Master of Science degree in experimental psychology at the University of Central Oklahoma in 1997. Working in the Department of Psychiatry and Behavioral Sciences, Dr. Halford authored or co-authored 4 peer-reviewed manuscripts and 24 published abstracts and poster presentations at Southwest Psychological Association and Research Society on Alcoholism. After a brief 10-month absence doing clinical trials research, Dr. Halford returned to sections of genetics in the Department of Pediatrics. He worked there. His work there began in the development and writing of research grants, but quickly he moved into the area of mentoring and education, which he had begun before leaving the Department of Psychiatry. While working full-time in the section of genetics, he completed his doctorate degree at the University of Oklahoma in the area of health and intercultural communication. Dr. Halford has been active in the areas of education and training, development, and directing the Summer Scholars training program for medical and professional students, and developing and teaching a course in clinical research methods for Masters of Science in Genetics Counseling. In addition to these, he provides lectures and seminars and research methods and serves as a research analyst for many research projects across the Department of Pediatrics. His personal research after leaving psychiatry has been primarily in the areas of promoting awareness of genetics concepts and understanding barriers to accessing genetic services among underserved populations, working especially with Spanish-speaking Marshallese populations, as well as Arabic students and Native Americans. This, together with his other research in the Department of Pediatrics, has resulted in 17 peer-reviewed manuscripts and numerous regional and national presentations. So without further ado, I would like to present to you Drs. Marza and Dr. Halford. Thank you very much for presenting for us today.
Am I going first?
I'm fine with if you want to go first, or if you want me to go first, however you want to do it.
Okay, since I'm going to lead into your talk, let me go first, and then let's see. Can I share my screen? Share screen 2. Let's go here. Oh, and I have to share first. Share. Can you see my screens?
Yes.
Okay. Let's go back to. First off, I'd like to say I have nothing to disclose in this presentation. I'd like to thank you for this opportunity to come and present. I was happy when Dr. Brand contacted me and invited us to make this presentation. As I say, it's been a number of years since I've been active in the Department of Psychiatry and Behavioral Sciences, even though I sometimes give talks over there, but I haven't been able to work directly in that department for quite some time. As my presentation here today, what I would like to talk about, why is this? Oh, there we go. I want to introduce the idea of how a person's basic genetic makeup can impact their response to alcohol and substance abuse, and some basic concepts of genetics. Dr. DiMarzo will then go further and provide some detailed information and explanation about the genetic changes, specific genetic changes that lead to and characterize specific genetic disorders. As the introduction suggested there, my interest in the area of the biological basis of behavior began in my work with in psychiatry under Dr. Sarah Jo Nixon and Oz Parsons at the Oklahoma Center for Alcohol and Drug-Related Studies. Of course, this is not a new area. Dr. Benjamin Rush, back in the 1700s, had written that why is it that some people who drink every day, drink heavily, can drink for years and years and not really have any problem with alcohol or drugs, or excuse me, not have any physiological problems associated with alcohol. But then other people who drink only mildly and for fewer number of years can sometimes have problems such as dementia or liver disease when they don't drink nearly as much as some of the persons who live their whole lives and don't have those sorts of problems. One of the basic ways of understanding this issue has to do with creating animal models of various health conditions such as diabetes and blood pressure, high blood pressure, or psychiatric disorders such as alcohol and substance abuse, schizophrenia, bipolar disorder, those sorts of things. So either research will take animals and expose them to higher levels of alcohol and drug use or drugs to see what impact that has on physiology and behavior of the animals, or else they'll develop through genetic manipulation, they'll develop specific strains of animals that are predisposed to or have psychological or physiological health disorders. When I first made this presentation back in 2016 or 17, what I did was start with some animals that exhibit behaviors that seem to be consistent with what we see in humans. First of these here is a new one that I found in doing a social media search a couple weeks ago, and that is cocaine bear. Now, I've heard about this meme several times, but I don't know the specifics. Apparently, some bear somewhere stumbled across, got a hold of some cocaine, and there are videos and information about the bear's behavior in response to receiving this particular cachet of drugs. There are a number of others available. It's not very hard to find if you search the internet for videos of animals behaving badly under the influence of alcohol and drugs. There are quite a number of those available everywhere you look. Now, back in 2016 and 17, when I was doing a lot of this kind of work, one of the stories that turned up in the news was about a pony in England that escaped from his stable, wandered down the street into an orchard and got hold of a pile of rotting and fermented pears, which he ate a whole lot of, became dizzy, and eventually fell into an empty swimming pool, and he had to be rescued by the local fire department. Another story had to do with some wax starlings that found holly berries that had fermented, and like out of a scene from an Alfred Hitchcock movie, started dive bombing and attacking pedestrians and people in the town. There are a number of many other stories of this sort relating to bees and elephants and bats and all number of different species of animals. Concerning primates, there's some stories written about some chimps, because in Uganda, where alcohol, because of Islamic traditions, is very difficult to get, various moonshiners had created some stills and were storing alcohol in storage sheds out in the countryside, and the chimps had identified these and were breaking into these stills and stealing the alcohol and getting drunk. And there were some baboons in South Africa that were breaking into vineyards and consuming fermented grapes. And the people cultivating the grapes would have to load them up in wheelbarrows and haul them off the next morning. What all of this suggests to us is that ethanol, being one of the oldest and most widely available energy sources in nature, has resulted in many animal species, not only animal species, but also plant species and also bacteria, a wide range of species being able to metabolize an alcohol and form. The gene responsible for this is the alcohol dehydrogenase gene, the ADH gene, which is the primary genetic mechanism that allows for the metabolism of alcohol. 7 variants of the ADH gene occur in humans. I'm not going to go into this in great detail because Dr. DiMarzo will be speaking of this later. But there are variants in those genes, some of which are protective against alcohol abuse and dependence. Some increase the risk of alcohol abuse and dependence, but these gene variants occur at different rates in different racial and ethnic populations, so it's not equally distributed across the human species. And the variants increase or decrease the risk of developing alcohol and alcohol-related problems, but which, if we want to use genetic language, means that it's polymorphic, meaning different variants change or influence the risk of developing alcohol-related problems. But it's also polygenic in the sense that not only the alcohol dehydrogenase gene, but other genes as well, in the human genome, increase or decrease the risk of developing alcohol-related problems, which makes it polygenic. Beyond that, it's not just the presence or absence of these risk influencing variants, but the presence or absence of those variants in the context of environmental factors, which influence their expression, that help to determine whether a person has or doesn't have alcohol-related problems, which makes it not only polygenic, but the word is epigenetics, or epigenic in this sense, meaning a confluence of not only genetic factors and biological factors, but also environmental ones. Now, of the genes that influence alcoholism in particular, about 50 to 60% of alcoholism can be attributed to these genetic factors. We spoke a little bit about alcohol dehydrogenase, but 2 other kinds of genes that are influential are the dopamine receptor variants, which affect the reinforcing properties of alcohol, how good it feels to drink, or a person's individual experiences under the influence of alcohol, and the GABA receptor variants, which affect inhibition in the frontal cortex, as well as drug-seeking behaviors, a craving for alcohol, alcohol withdrawal symptoms, and so forth. So it's the interplay of all of these different factors that influence whether or not a person does or doesn't develop alcohol-related problems. Another set of presentations that I developed after completing my dissertation, which was on cultural aspects of alcohol consumption, was what's sometimes called the firewater myth. A number of years ago, I was presenting to, or I was interviewing Native Americans about their alcohol use, and I was at a Native treatment center, and I'm interviewing one person. I asked them the question, when do you think you first developed a problem with alcohol? And he said, in the womb. And I said, do you mean like your mother was drinking alcohol, and so your alcoholism is based in what happened to you while your mother was pregnant? And he says, no, I'm an alcoholic because I'm Native American, and all Indians are alcoholic. I interviewed a woman who was in another treatment center and asked her about that, and I asked her about her alcoholism. She said she was an alcoholic, and I asked her when did she first begin using alcohol. And she says, "Oh, I've never in my life have I ever had alcohol to drink." And I said, "Well, why do you think you're an alcoholic then if you haven't ever had any alcohol?" And he said, "Well, because my mother was an alcoholic, and my grandfather, and aunts and uncles, and so forth. And so I'm certain that if I ever took a drink of alcohol, then I'd be an alcoholic too." And this notion, of course, is associated with the wide cultural belief that Native Americans are biologically predisposed to become alcoholic. And through the course of my dissertation and the presentations I've made since, I've attempted to overcome, or at least to challenge this particular stereotypic belief. And of course, this is not specific just to Native Americans. There are other cultural groups that tend to be seen as being particularly prone to alcoholism. So say Russians or the Irish or Italians have been accused of this particular stereotype. So the notion here is that it's more about biology than any cultural aspects. Perhaps, well, not perhaps, but most certainly a better approach would be to look at this in what's called a biopsychosocial model, and that is to recognize the fact that you have interplay between genetic and biological factors, cultural aspects, and personality characteristics, things that happen to the individual and how they've responded to that those events in their lives. Those three things in combination are a better explanation of how certain people develop problems with alcohol and others don't. On the more physiological area, we certainly know that things like age and gender, and other biological characteristics such as weight and height and the hormone levels and amount of exercise a person gets, but also things like the concentration of alcohol, whether one drinks low alcohol content beers or wine as opposed to hard liquor, whether one drinks either alcohol in combination with foods or drinks on an empty stomach, and their history of drinking over time. All of those things affect the metabolism of alcohol, and that's widely understood. One of the things that's less understood on the psychological side are things like context-specific tolerance. And a number of years ago, there was a lot of focus on people who had overdosed and probably died as a result of using drugs in various places. You hear stories going back as far as Marilyn Monroe and other Jimi Hendrix, a number of famous musicians and movie stars and so forth who died of drug-related overdoses. And sometimes you'd hear that they would die in unusual places like hotel bathrooms and so forth. Research looking into some of that suggested, the hypothesis was they used either a purer form of the drug than they normally took, or else in ways that, or they took more than they normally would take at any given time. But research seems to suggest that the context makes a difference. If you're going to, usually if people are going to go out and drink with their friends, they think about what it's going to be like. Oh, it's a New Year's party. We're going to go drink and dance and have a good time. And it's going to be really fun and all of this. Psychologically, we prepare ourselves for receiving alcohol or drugs, and that psychological preparation changes how we respond physiologically to the drug. So if we're in the right frame of mind, then we're able to better metabolize or biologically deal with the drugs when they enter our system. But if we don't prepare, if we're not properly prepared, then our body is not ready to receive that. And so even receiving the same dose we get in some other context may cause an overdose or uncharacteristic behaviors in response to the alcohol. On the cultural side, there are things like, as I mentioned before, there are certain countries for religious reasons or whatever that alcohol is not very widely available. And so people drink in ways that are different than people who drink in societies where alcohol is normative. That affects the accessibility of alcohol, the kinds of alcohol that are available. But cultures also differ on what sort of behaviors are acceptable in terms of alcohol. Some of the questions that we ask about alcoholism is, do you ever drink alcohol in the mornings? Do you ever drink alone? have you ever been drunk in public? Some things like public intoxication and drinking alone and hiding your alcohol consumption play into diagnosis of alcohol-related problems. Well, there are certain countries in the world, thinking back to what I said about Italy and Ireland, where it's pretty common for people to drink and become alcoholic in public. And so those things are not necessarily seen with the same sort of light as they're seen here. Things like getting drunk or drinking alcohol for the purpose of getting drunk, or getting drunk and getting into fights and things are more acceptable in other cultures. And so if you want to understand overall the way that people relate to alcohol, then those cultural aspects have to come into play too. So in some, you have the biopsychosocial model, and in order to best understand the impact of alcohol abuse and alcoholism, you have to consider the physiological characteristics, the cultural and the psychological ones as well. Returning now to the genetic side of things. If we want to understand the genetic impact of alcohol and other substances on persons, animal studies. I said at the top that the one best ways to do that is by looking at the behavior of animals and seeing how changes in genetic characteristics affect animal behavior. Another way in humans is twin studies. Dr. DiMarzo will speak of this quite a bit more in a few minutes. And finally, the other side is taking good family health histories. As with any other health measure, the best way to find out about a person's situation is to ask some questions about what's happened in their family. If we want to know about cancer, we say, well, has anyone else in your family had cancer? What sort of cancer did they have? How old were they when they were first diagnosed? What was the outcome? Did they survive this, or did it kill them? Did their cancer ever recur? Those are the sorts of questions that any physician would ask if you want to know about some health condition. And certainly we would do the same thing in pursuing alcohol and substance abuse. Now, in twin studies, as I say, Dr. DiMarzo will talk more about this later. I'll just say here that there are some difficulties in doing research with twin studies. Number one, because of adoption rules, it's very difficult to identify twins that have been adopted out, and twins separated at birth is the best approach if you want to eliminate social and cultural factors. If children are raised in different households, then the income of the outcome of those cultural factors and social factors are averaged out because they're raised in different environments. But with closed adoptions, it's very difficult to obtain access to them. And then HIPAA rules, privacy rules affect our ability to obtain information about other individuals beyond the patient that we're seeing at any given time. This also affects family health histories. I remember back when I was doing alcohol research, we had a family tree that we passed out at treatment centers and asked people to fill out. And then I would go around one-on-one and ask them specific questions about drinking patterns and substance abuse patterns in different family members. And oftentimes I'd come to someone and across the top in big bold letters, it'd say, "None of your damn business." We see this anytime we ask family health histories. It's easy enough for patients. They're usually willing to talk about their own behavior, especially if it's stigmatizing behavior or things that has happened in their own lives, but they don't think it's anybody's business to talk about other families' substance abuse patterns. But also, because of the stigma attached to that, it's also difficult for them to know. Do they know how old certain things happened to other people, when they were diagnosed with this or that, what happened to them, what age they died at, and that sort of thing. Even in close-knit families, that sort of information is hard to come by. And so there are limitations in what we can find out through family health histories. And so there's a lot that can be done by looking at genetic factors. I mentioned the term epigenetics before, and this is the notion that while genes are the most basic element of physiological processes, our genes determine a lot of our characteristics, how long we're going to live, how tall we are, what health problems we may or may not have, how good we are at various sorts of physiological activities and so forth. There's always, from the very beginning, physiological characteristic or physiological things, life events, environmental contexts, all of those things can influence our genes. They can, the different process we'll talk about here in just a moment, having to do with the activation and silencing of genes that affect how those genes are able to function. But also the characteristics we talked about before. If you think about how tall a person is, genes may specify a range of how tall a person may develop in life, but various life events, things like disease and diet and how much exercise a person gets, will ultimately impact how tall a person actually develops. It will be somewhere in that range that their genes specify, but where they fall within that range, the environment makes a big difference on how that falls out. And of course, this interplay between genes and environment really identifies the individual as unique in this same way. In this way, they would be very different than even identical twins. Another concept in genetics is, and I've mentioned this a little bit before, and that's the difference between single gene disorders and multigenic disorders. And the simplest way, you may have something, a condition that one gene makes the determination whether or not a person has or doesn't have a characteristic. So the perfect example here would be something like eye color. The only way one can have blue eyes is if you inherit the gene for blue eyes from both parents. So it's a recessive gene, which means you have to have both the gene from your mother and the gene from your father, both supporting blue eyes in order for you to have that characteristic. A dominant gene would be if you inherit a gene from one of your parents for that characteristic, then you would have that characteristic. Multi-gene disorders are harder to identify because, again, this involves the interplay of sometimes dozens, sometimes hundreds of genes across the human genome before you have or don't have that disorder. So, for instance, a number of years ago, well, in the last 3 years, there was an article published suggesting that there are as many as 8 new genes have been identified for schizophrenia. And so it would be the interplay of all of these different genes, which would ultimately affect whether or not you were likely to develop that disorder. The same is true for a number of, especially psychiatric disorders, which tend to be more complicated and involve the interplay between more and more genes. But then even things like diabetes and cancer, there are sometimes hundreds, sometimes thousands of genes that are involved. The other idea to consider here is the notion of penetrance. People like to say, well, you know, if you smoke, you're going to get cancer. And the fact is, as was said earlier about alcohol, if you drink a lot, it doesn't necessarily mean that you're going to become an alcoholic and have alcohol-related problems. But there are some genes that predispose for alcoholism. The same thing would be true for cancer. If you have certain cancer genes, you're much more likely to develop lung cancer if you smoke. So penetrance is the notion that there's a large environmental influence that helps to determine whether or not you have a certain disorder. So, a high penetrance gene like the BRCA breast cancer gene, if a person has that, then their likelihood of getting breast cancer is really high, approaching 100%, especially if a person smokes. Whereas other genes, if you have certain cancer genes, then having that gene may double your risk, but doubling your risk would only amount to going from 1 in 500 to 1 in 250 chances of having that particular form of cancer. The notion of imprinting is, even if you have genes, oftentimes the gene that you have from your mother and the one that you have from your father, biologically, you don't need both of those genes to function. In fact, you need one of them to be silenced or imprinted, which means to coat that gene in such a way that it's not able to be biologically active, to code for proteins. And so one or the other of those genes gets silenced. Sometimes you get too many of genes. For instance, in Down syndrome, where the whole chromosome 21, you have 3 copies instead of 2, which means you have 3 working copies of certain genes, which produces far too much of certain proteins, which result in the typical Down syndrome phenotype. So you can either have an overexpressed gene or an underexpressed gene, and to some degree, imprinting is the biological process that tends to limit that, which occurs naturally, but can also cause problems to develop after birth. I'm going to skip over a couple of things here because, as I said, I had a problem with my PowerPoint presentation because the power went off here and some things are included that probably shouldn't be. One of the things, as I've said, is this notion that where disease, conditions, and diets can dramatically affect things like your adult height and other physiological characteristics. In the same way, certain physiological and psychological experiences can impact your genes, can impact this this notion of imprinting. So a person who drinks heavily, it can cause a change in how the genes are expressed in the individual, which in turn affects how the body metabolizes and responds to alcohol. So you have this circular process where DNA affects your physiological response to alcohol, and the environment in which you consume alcohol and the amount of alcohol you consume in turn then affects your physiology, which can in turn affect your genetic functions, which can in turn affect your response to alcohol, and this goes around and around in circles. To look at this in a more concrete way, this is it's not appropriate necessarily to say mother, but this is an adult and the clone of that adult cat. You can see here in this particular photograph that the patterns of fur color are different. And more than that, the colors themselves are different. Now, if only the DNA mattered, then you'd expect to see identical cat. The notion of taking your very loved pet and cloning them and having that pet again doesn't hold up. The first cloned pets, pet owners paid upwards of $50,000 to have their pets cloned. And when they got them, they were disappointed because it wasn't Scruffy 2, but some dog or cat that looked somewhat like Scruffy and may or may not have acted the same as Scruffy, even though identically their genomes were the same. Beyond the way they look, here's a pair of dogs, the host, if you will, and the clone. Identical, completely identical DNA. Not only is the pattern of fur color and different, but the behavior is different. In this case, the study focused on behavior. One dog was certainly more attentive, or more responsive, responded differently to presence of new stimuli. In other words, they responded differently to strangers. One was more physically active than the other, more curious than the other. So even these psychological characteristics, even though they were raised as much as possible to be identical, and their behavior was certainly not the same. So not only does the environment affect appearance, but it also affects behavioral characteristics. And finally, the same can be said of human beings. If you see sets of identical twins, they're not, you can see from this picture, hair color is not the same, height is not the same. We could go further and say their behavioral characteristics were certainly different between these two individuals. And this is just one example of literally hundreds of thousands of pairs of identical twins that could be compared and found to be quite different in many, many ways. The take home I'd like you to consider from this presentation is that if you say that genetic characteristics define and determine physiological traits and even psychological traits, the only point when you could say some particular trait is entirely genetic would be at the moment of conception, because from conception on, then what happens in utero, what happens in development after birth, diet, exercise, psychosocial influences, all of those things not only influence the behavior, but it also influences the gene. So in as much as our characteristics are genetically based, you can only say that it would be purely genetically based at the moment of conception. Anything after that is heavily influenced by physiology and environment. I probably exhausted my time now. I'll pass this along to Dr. DiMarzo.
All right, let's see if I can get this working a little bit better. All right. Share my screen. Can you guys see my screen okay?
No.
I cannot see your screen.
It's not showing up. Okay, let me try this. Okay, is it working now?
Yes.
Yes.
Perfect. All right.
I wonder if you, yes, there you go.
So first things first, similar to Dr. Halford, I don't have any financial relationships. I don't have any disclosures. I have no conflicts of interest. You'll see some slides that I put in that I removed the lab logo and decommercialized it for the purposes of this, but I don't have any relationships or affiliations with them. And they're purely for educational purposes. So my objective for this was to review the currently known genetic associations for addiction, chemical dependency, and its biological neuropathways. Hopefully by the end of both of our talks, which are kind of meant to complement each other, there are some repetitive information, but hopefully that will just help solidify the most important information. And it's more complementary and building on each other. So hopefully that will give you a good awareness for the current known gene associations, make you aware of the currently available testing, which isn't much, but it's out there and it's pretty good for what it is. And kind of help toward the end here, wrap it up with its knowledge and how those test results can help you guys in terms of management of your patients. So to start off, why are we suspicious for a genetic component? Well, we know that substance use disorders often run in families. Is it hereditary? Is it environmental? Is it a combination of both? I really like that slide Dr. Halford shared, where it showed the combinations of, genetics, behavioral components, and that kind of Venn diagram that showed that there's significant overlap. Family history often offers some clues. Researchers have used family history and compared DNA sequences in family members to identify genes that are involved in addiction. They look for genetic components that are more common in affected people compared to unaffected people to kind of show for some linkage components. So the National Institute of Drug Abuse, or NIDA, has a division called the Neuroscience and has a division of Neuroscience and Behavior, and its advances in science and drug addiction through the basic and clinical biomedical neuroscience and behavioral research. The DNB's Genetics, Epigenetics, and Developmental Neuroscience branch supports research in genetics, epigenetics, and developmental mechanisms that underlie substance use, misuse, and addiction. Gene sequencing is an incredibly powerful tool, and it finds connection between known genes and disorders. And it's identified genes that may have been overlooked or were previously unknown in previous, when we did the Human Genome Project and subsequent sequencing. Recent advances in sequencing technology has helped advance our knowledge of the genetic basis for many diseases, such as autism, neurodevelopmental disorders, and subsequently addiction and mental illness. So what do we mean when we talk about, so first of all, what do we mean when we use some of these terms? We talked about a single gene disorder, and this is a gene that's caused by a pathogenic variant in a single gene. And so when we think about these, think of like cystic fibrosis, we know is caused by changes in the CFTR gene. Marfan syndrome, we know is caused by changes in FBN1. And we know that it's multifactorial inheritance. We know that this follows a specific either dominant or recessive inheritance pattern. Multifactorial inheritance, when we talk about this, we often refer to it as complex inheritance. There's more than one factor in a complex trait or health problem. We think about these like birth defects like cleft lip, cleft palate, chronic illnesses. Genes are often a contributing factor, but other things that are not genes can also play a part. So neurodevelopmental conditions such as autism, a lot of the psychiatric disorders fall into this category. We've talked a lot so far about epigenetics. Epigenetics are functional, sometimes inherited changes in the regulation of gene activity or their expression that are not necessarily dependent on the gene sequence. So something like methylation or histone modification can be a significant component in epigenetics. So addiction in general is not considered a single gene disorder. It follows a multifactorial complex inheritance pattern with evidence of epigenetic components. We usually talk about nature or nurture, and now the thought process is nature and nurture, because we know, as Dr. Halford very eloquently laid out, that it's not a person's health is not is the result of a dynamic interaction between genes and the environment. So when we talk about an addiction gene, what do we mean? A variation in gene. There are variations in DNA sequence that make someone more or less vulnerable to addiction. And these variations can cause an individual to respond to drugs and medications differently. A vulnerable person may have a high preference for a particular substance or experience extreme withdrawals if they try to quit. A less vulnerable person may feel no pleasure for that drug. That makes other people feel very euphoric, so they may have a less effect from the substance. A more vulnerable person may have more severe withdrawals if they attempt to quit, and vice versa. So many genes contribute, and many genes may be contributing and influencing. We will likely never find just a single gene that causes addiction or is responsible. It's not like cystic fibrosis or PKU, where we know the gene. Substance use disorders is a complex trait that's influenced by multiple genes, plus environmental factors acting on the gene's expression. So it's more than just running a simple test. And also people with substance use disorders also have different genes that can be contributing. So it's what we call genetically very heterogeneous. People who share certain high risk gene variations may or may not develop substance dependency. And this is where he mentioned penetrance. This would be incompletely penetrant. So not necessarily if you have a gene change in a high risk gene, does that mean that you're going to develop dependency? And this may lead to hesitancy in someone wanting to get tested. This makes finding a precise genetic cause tricky and results difficult to interpret from the test findings. Researchers have estimated that genetics accounts for somewhere between 40 to 60% of someone's risk of developing dependency. So where do we get all this data from? There's twin studies, there's adoption studies, we have linkage analysis, and there's newer studies have come from what we call GWAS data. So adoption studies, you have an adopted child and we look at them when they're raised. There's different types of adoption studies. These studies have attempted to kind of tease out the importance of environmental versus genetic factors in development of phenotypes and disorders. These are considered a key tool in behavioral genetics and other related fields. So when we look at twin studies, psychiatry has really loved twin studies. We know that there's from a genetic standpoint, two types of twins, monozygotic and dizygotic twins. Monozygotic twins are genetically identical. We are assuming that they have 100% genetic match. And we look at them if they're reared in the same home or if they're reared apart. And that looks at the difference of the environmental impact. If we look at dizygotic twin pairs, we're assuming on average 50% of genes changed. And we look at them reared together versus reared apart. And we're assuming the role of the environmental impact would account for the difference that we see. So some studies are looking at the difference between monozygotic versus dizygotic twins and assuming that that would calculate the difference. So it looks at the risk of receiving the diagnosis in the same versus is it the same amongst twins versus non-twins? And is the diagnosis the difference between monozygotic twins versus dizygotic twins? And the difference in experience and how the environmental impact changes versus the genetic impact changes. So that makes a lot of assumptions in terms of concordance rate and the statistical significance between those is thought to be the difference, and is the statistical significance attributing to the genetic factor. So that's how twin studies run. Linkage analysis. This is a very powerful tool for mapping a chromosome location. You can see here, I don't know, can you guys see my cursor?
Yep.
Okay. So what you do is we draw the pedigree, we map, we track who's affected, and then we look at different gene markers and we try to map it to the certain loci. And then it's a statistical method for mapping the heritable trait to genes to their chromosome location. And it attempts to locate the disease gene to the causative gene by identifying the marker of the known chromosome location at a co-inherited trait of interest. So we figure out where it is based off of the shared genes on the pedigree amongst the affected individuals. Now the new trend is GWAS studies. GWAS stands for Genome-Wide Association Studies. It's an observational study of a genome-wide set of genetic variants in different individuals to see if variants are associated with a trait. This method studies the entire genome of a large population searching for small variations of small nucleotide polymorphisms, or what we refer to as SNPs. Each study looks at hundreds of thousands of SNPs at the same time. And so what we do is there's data collection amongst a large population cohort. We do genotyping, we look for different SNPs, and then we kind of compile it to see, do these SNPs map to different, what is the genotype at the SNP at a specific location. The SNPs can help research pinpoint a gene that are likely involved in the disease development for that trait. As you can see here, and we kind of see how the components track amongst the different populations. And I'll talk a little bit about what the issues are with that based on the population cohorts in a minute. So what does all that data find about addiction and alcohol dependence? First of all, we have some contributing genes. Alcohol dehydrogenase is one of the big ones. The ALDH2 is part of the major pathway for alcohol metabolism. There are two major ALDH isoenzymes in the liver. The first is cytosolic, the second is mitochondrial, ALDH1 and ALDH2. ALDH2 is responsible for this acute alcohol flushing reaction. One of the isoenzymes is responsible for this acute flushing reaction, predominantly seen in the Asian population. There's a particular variant, GLUT405 lysine, which is referred to as ALD2*2. It's the best characterized genetic factor influencing alcohol drinking behavior. Those with this atypical allele are at much lower risk for alcoholic liver disease due to their sensitivity to alcohol intoxication. So basically, you develop an adverse reaction when you drink, you're less likely to develop adverse drinking behaviors. Those with ALDH2*2 experience nausea, facial flushing, headaches, rapid heart rate with alcohol consumption. So you can see here's the pathway, converts alcohol to acetic acid. And when this is malfunctioning, you can develop carcinogenesis. When you have this malfunctioning, you have facial flushing, tachycardia. Again, it affects 450 people worldwide, but is seen in 7.3% of the population, but mostly concentrated in Asia. Okay, I'm going to try to be conscious of the time, so I'll kind of go through these a little bit quickly. DRD2 is in the dopamine pathway, and it is a postsynaptic dopamine neuron, and it is involved in the reward pathway, and it signals dopamine, the dopamine receptor. The presence of the A1 allele, it was correctly identified in 77% of alcoholics, and its absence was correctly identified in 72% of non-alcoholics. So the A1 allele is more common in people addicted to alcohol, cocaine, and opioids. DRD2 is also associated with other mental illnesses, including schizophrenia, schizoaffective disorder, major depressive disorder, and conduct and emotional disorders. So you can see here's the dopamine pathway. It is part of the reward system in the frontal cortex, controlling compulsions and mood, memory, processing. And here's just the dopamine receptors. There's also OPRM1, and this is part of the opioid receptors, and that's its primary site of action. We know that there's 3 different types of opioid receptors. This is the mu receptor. And this is a candidate gene for pain perception and response to opioids. And this variants in this gene have led to susceptibility to substance dependence. So we can see here, we know that T is the common sequence, and cysteine is the SNP that we see. And you can see here that in the general population, you see phenylalanine, and we see that much less frequently, and we see the SNP more present in the cocaine and heroin addicted population. And here's just a reminder of what we see in the mu receptor in the central nervous system and the descending transmission of pain. I'm trying to remember. No, not that one. Okay. Acetylcholine pathway, the nicotinic acetylcholine pathway receptors, CHRNA5 and CHRNA3. It's had a cluster of influences, and these alleles have been associated with nicotine dependence. So if you have 2 copies of this allele, you're twice as more likely to become nicotine dependent as people with the normal, the wild type allele. And this is also associated with increased risks of lung cancer, peripheral artery disease, because as Dr. Halford pointed out, the habituation also has influence on other health risk factors. MIR-212 also has post-translational gene expression that plays a key role in the vulnerability to cocaine addiction. This is a hodgepodge, and I know this is a long list, but this is mostly for reference. We have the DRD1, DRD3, DRD4, which also relates to other dopamine pathways that are linked to alcohol dependence. The GABRB3 is in the GABAergic pathway that is also related to mood disorders and different alcohol dependence and other substance abuse disorders. MAOA is also leading to impulsivity, psychosocial behaviors, and substance abuse disorders. Now, how does this translate into what we're going to do day to day? Well, we know that certain pathways can help target certain medications. So this is where what we call precision medicine or pharmacogenomics comes into place. So when we look at this, I got this from one of the companies. This is a sample of how they're, well, this isn't exactly what the reports come back, but when we look at certain, we know that these different medications are involved in certain pathways that certain drugs target, right? We use medications that target dopamine pathway. We use medications that target the nicotinic pathway and the serotonin pathway. So if we know that your patients have polymorphisms that are going to affect how we metabolize, if you're a poor metabolizer, ultra metabolizer for certain medications, how does that affect treatment? So precision medicine looks at these different pathways and says, do you have SNPs or polymorphisms on these pathways that could help your medication, help you target your therapeutics? So personalized medicine is important. Precision medicine is also referred to as personalized medicine. This uses the information about the person's genome to prevent, diagnose, and treat disease. It eliminates this one size fits all. Okay, I'm going to start everyone on an SSRI. I'm going to start everyone on this treatment protocol first, and this one size fits all approach, and tailors it to help prevention strategy. And it customizes care for these differences between individuals. The goal is to avoid serious adverse drug reactions by giving people a medication they might be a poor metabolizer for. It avoids drug-drug interactions. It replaces traditional drug choices and a dose, trial, and error treatment plan. It optimizes the drug choice based on a unique genetic profile, and it helps decrease the overall healthcare approach. The benefits are it increases safety, it improves overall outcome, it decreases adverse drug effects, you get patient relief, improvement, and it could potentially be life-saving. We know that certain medications come with labeling on it due to in order to prevent potentially life-threatening reactions. It lowers medication expenses due to multiple trials. On average, they say that most patients go through about 3 to 4 different antidepressants before they find one that they like, or the one that is effective for them. That leads to lower healthcare costs and a selection of the appropriate medication, and it maximizes drug efficacy. And it also what the main goal is to protect from non-productive toxic medication treatment. So this is what on the right-hand side, this is what you will see as a potential lab report. It'll say these medications based on the pharmacogenomic profile for your patient, we will use as directed. These different medications you are based on the metabolic profile should use with caution. These you either should use with extreme caution or with very close monitoring. So these are how the reports come back based off of whether they're normal metabolized or slow metabolizer. So this has led to some pretty good results for some of our patients. The practical utility, it's pretty easily attainable. There are some centers that use this as, pretty standard of care. You get results pretty quickly, usually in about 2 weeks. You can avoid toxicity and overdosing. This can decrease the length of time to effective treatment and avoid multiple medication trials. But it is, even though it's easily attainable with pretty rapid results, it's rarely utilized. In a 2021 trial, which now that seems pretty dated, only 4% and 10% in a Medicaid and Medicare cohort were used, despite that all participants selected were taking medications that contained labeling information that said it was indicated. So that's pretty significant. So that, yeah, it was pretty, I found that pretty strikingly low. I thought that would have been higher. So that's one component, the pharmacogenomic studies. Another study is the actual diagnostic. So that's treatment and management. In terms of what we're going to do for diagnostic purposes, polygenic risk scores. Polygenic risk scores have been pretty significant in helping us manage patients and give them an idea of what their genetic susceptibility and risks. They came up with this thing called the GARS risk score. And this is a molecular genetics has been kind of important in linking aberrant behaviors to individuals. Polygenic risk scores have been utilized for a while, probably a couple of years now in cancer, where if we don't see someone with an individual gene that we know is associated with a risk, we compile all the genes that we know have low risk and we come up with a composite score. So for here, in spite of search to uncover a specific candidate gene, these risks have not been very conclusive. They haven't been easily replicated. So we know that these candidate genes, the genes that we've talked about, DRD1, DRD2, MOAA, COMT, these can come up with personalized solutions and clinical outcomes. There have been 10 reward genes associated with risk variants that have been used to create this risk assessment tool called GARS. GARS significantly predicted the severity of alcohol drug dependency. It genetically identified a risk for all reward deficiency syndromes, or what they refer to as RDS behaviors, especially in a compromised population. So let's say you have a significantly strong family history of addiction. They're proposing this as a frontline tool to assist municipalities to provide better resource allocation. So putting this in a particularly high-risk area. Genomic testing such as GARS can improve decision-making and clinical interaction. So GARS can be utilized to specific neurotransmitter pathways to identify where the breakdown may be. While having this knowledge doesn't necessarily identify a therapeutic approach, it has important clinical value. It can help with alleviating denial or guilt. It can help provide assistance in risk severity decision-making Can I treat this person outpatient? Can I treat this person inpatient? Is this someone that I necessarily want to prescribe pain medication to? It can also help in determination of pharmacogenomic medication monitoring. And for example, we know that the DRD2 gene diminishes naltrexone clinical effectiveness. So that might not be someone you want to use opioid substitution therapy for. And it can provide insight in terms of risk of relapse. A polygenic risk score looks something like this. We know that it represents a total number of gene variants that an individual has to assess their heritable risk for developing a particular disease. We can sometimes, some reports frame it as a percentile in terms of the population, some present it as a risk ratio or an odds ratio, and some present it as a 10-year versus a lifetime risk. We talked about how it applies to different population cohorts. Most polygenic risk scores are based off of a Caucasian European population. So be careful applying this with some minority cohorts because it might not necessarily translate as well. So that is a little bit trickier in terms of its relatability to different minority groups. This has some barriers to performing different than pharmacogenomic testing. It tends to be slightly more costly. It's not as widely available. The labs might not be contracted with your insurance. You might have some direct to consumer labs offering this that might give you some weird results. And it's not as universal in terms of what labs are what genes are included. And the content might be slightly different. What are some ethical dilemmas? Well, the formatting makes different sharing from different institutions a little bit different, and you might not get all, it makes it a little difficult. You might not get all the same information. There are significant privacy and security concerns, and genoprotections don't always protect information that is contained in this for certain people. Some individuals who are unaffected but have a significant family history don't want to undergo this because they're worried about stigmatization in terms of, I'm someone who doesn't have addiction, but I have a family member who has it. Do I want to undergo this risk assessment and have that label if I currently don't have a problem? And would that necessarily lead to me having issues with getting certain types of insurance protection? So there's some significant ethical dilemmas that go around that. Now I'm coming to the end here, so thank you for bearing with me because I know I ran a little bit over. Putting this all together, we do have some addiction and reward deficiency syndrome behavior issues. We have genetics and epigenetics coming together. We can use precision medicine, GARS data, to come up with preventive strategies, treatment strategies to attenuate the disease and come up with a treatment and management plan. And so we have treatment responses that are very heterogeneous with multiple factors to consider in terms of our management. Many individuals fail to respond. Patients show various clinical responses and side effects to the same medications, even at the same dose. Using genomic and epigenomic approaches can be a way of delivering a new potential drug targets. Some of the examples that we've come up with using these epigenomic data and pharmacogenomic data is coming up with Topamax and Zofran and Baclofen and gabapentin. These have been tried. The results weren't statistically significant, but these are different ways that we're trying as we're discovering new pathways, and new trials are coming up on the horizon. So customizing approach to clinical care is what's basically coming out of this. So in summary, there's promising advances in genetic technology, and our understanding of addiction and mental health is rapidly advancing. Testing has helped us better tailor our medication, and it remains to be seen how this will pan out in terms of therapeutic approaches, but it has shown great promise. So stay tuned. And I put this on there because this is the most prevalent addiction nowadays, is addiction to technology. And that's all I have. Any questions?
Thank you both so much. This is Kelsey. We both really appreciate you coming on and giving us this presentation today for the attendees of the chemical dependency seminar.
Oh, my pleasure. I hope you guys enjoyed it, and I hope I didn't drag on too long.
Not at all. We appreciate it so much.
Yeah, I would also like to say thanks again. Yes, we did very much. I always enjoy making presentations here, and I just hope it was useful to your audience.
You both. We really enjoyed it. Well, if no one has any questions, I think we're going to conclude here. So
Yeah.
we will end it. So thank you so much.
Thank you, and thank you for putting this all together.
No problem. All right. Bye-bye, everybody.
Bye.